we have one that I think that is good to share this. Oh, you boleh copy paste kepada sini.
Here.
Yeah, bawah itu copy and then ambil itu ke
I'll just call it an aberration. It's that Frankenfish is what we call it.
Frankenfish. Not Franken? Fish that they're talking about? Well, this is it. It's a binary fish, and it's got a lot of people doing a lot of things.
It is so unnatural.
It's the first bioengineered animal ever approved for consumption. And after 30 years of debate about whether we should eat it, it's now on its way to your dinner table. And it may forever change how we produce food. We're here outside of Aqua Valley's Indiana facility. Just last week, they received their first shipment of Aqua Advantage salmon eggs. Now, these eggs, when harvested, will become the first genetically modified animal ever approved for consumption in the U.S. So, these are the eggs. They don't look like much, but they're the culmination of decades of research and debate over whether genetically modified animals have a place in our diet.
We're going to see a 2 billion person increase in the population in the next 30 years. So we're going to feed 28% more people. So you start with that premise, and then you think, how is seafood going to play a role? Wild caught salmon is basically at full capacity. Other farm method is ocean cages, and they have their own set of challenges because those fish are exposed to climatic conditions, predators, disease.
And that's where Aqua Valley's fish come into play. 30 years ago, scientists figured out how to produce a salmon that grows in market weight in half the time as conventional salmon. To do this, they injected a growth hormone from a Chinook salmon into an Atlantic salmon, along with a hormone from the ocean fowl to ensure the rapid growth is constant. These super fast growing salmon have two big advantages. First is that they need a lot less food.
They have what we call an improved feed conversion ratio. And what that means is for every pound of feed that you put in, you get more than a pound of meat out. So if you think about cattle, it takes about 8 pounds of feed to create 1 pound of meat. With our fish, you put a pound in and you get like 1 1/4 pounds out.
Since they grow faster, Aqua Advantage fish require 25% less food than conventional salmon. The second advantage of fast-growing salmon is that you can simply produce a lot more of them, and that's what unlocks Aqua Valley's other upside. They grow their fish in what boils down to big tanks, but the facility is actually pretty high-tech. It has some huge upsides. Super tight controls on conditions prevents disease. Nearly all of the water is recirculated, so it's ultra efficient, and you can put them pretty much anywhere. The problem has been that it's so expensive. When you have fast-growing fish in there, the numbers start to balance out in a way they never had before. All of this may sound great, but it's been far from a cakewalk. Genetically engineered anything is rife with controversy. The longer of the market is the result of an approval process that has surfaced two consistent and pretty reasonable questions that arise when considering genetically modified animals. What if they get out and overrun native populations? And more prominently, are we sure they're safe to eat? At the Indiana facility, Pete, the farm manager, is the point man for making sure the first one remains moved.
We have, first of all, on a really large scale, we have geographical location. We're in Indiana. There are no native salmonids. Nevertheless, inside our units, we have a very high level of physical containment. Every possible escape route out of the facility, water discharge points, pipes, layers contain physical barriers in place that will prevent the passage of the fish. We do have numerous redundant layers.
And on top of the physical barriers, nearly every single fish is sterile. If one gets out, it can't breathe. So then the question remains, is it really safe to eat? After decades of asking the same question, the answer from regulatory bodies is yes. Why did they take that long? Well, since there was nothing else really like it, there was no process for it.
So in the case of animals, they didn't have a really good law. So they used the Federal Food, Drug, and Cosmetics Act.
This is Dr. Eric Hopkins from Virginia Tech. He was brought in as an expert during the process.
What they said is the transgene is basically a drug, is altering the structure or function of an animal. The traditional food, Atlantic salmon, it's not regulated at all for that. In fact, we learned a lot about the biochemical composition of Atlantic salmon because Aquabounty had to go into baseline research on what is conventional Atlantic salmon. Then they had to show rigorously that theirs was no different.
And ultimately, that's exactly what the FDA decided.
Basically, the way that FDA typically put it, Aquabounty salmon is Atlantic salmon. That's it.
With all the regulatory questions settled, Aquabounty has the green light to sell their salmon on U.S. shores. And now the looming question is, what does this mean for the industry? Does Aquabounty's long fight with regulators clear the path for more innovations like it, or does it serve as a warning to anyone else trying to bring bioengineered animals to market? Dr. Allison Davidanon from UC Davis appears as the latter.
I've worked in this field for almost 30 years, and there's one product that's ever got to market. I mean, how depressing is that? There's been a number of really useful applications that have been developed over the last 20 years, and they're all basically sitting on the shelf because no one has been able to raise the capital to bring them through the regulatory process. But the problem is that it precludes the use of that technology in food production and more concerningly in developing countries. The average first world consumer has no idea that their choices are actually kind of impacting the ability to use innovation in the developing world. I always find it strange to be like categorically opposed to technology. I think you have to look at it on a case-by-case basis, and I think most people want healthy and safe food and also to reduce the environmental footprint of our existence on Earth. And quite honestly, I can't think of an industry that more requires, innovative technology to reduce the environmental footprint of food production.
Conventional methods are not going to feed 2 billion extra people on this planet. And it certainly is going to happen without some pretty significant impact to the environment. We're proud of the fact that, we solved the problem and we use science to do it.
Okay.
Thanks for watching. If you like the future of food,
Very interesting, right? Discussion. So what do you think? You
Thank you for
are on
having
the
me.
side of from the lecturer of University Davis, or you are CEO of Aquabounty, which is they are proud to have like innovative bioengineered fish to solve the problems. But in terms of the lecture mentioned just now, how we have to think about the safety, the food safety, right? So what do you think? So do you think that, or you think that I'm okay, which one? It doesn't matter as long as I can eat salmon? Which one? Okay, so which one? Ayo, kamu rasa kamu di mana? Kamu rasa kamu di mana? Based on the video just now, what do you think? What is your opinion? For example, Indonesia, the government of Indonesia agreed to have this bioengineered animals in the market. So this means that you don't know whether, means that you have a 2 fish. You don't know whether this one is a GMOs or not GMOs. What do you think? Do you, what is your response to your government for this one? Come on, what do you think? Anyone? If your government that release this bioengineered in the market. company, they come up with a bioengineered fish just now. Okay? And then the government agreed to release in the market. Boleh dijual market. Jadi kalau sendiri, market kamu senang tidak tahu bahawa yang dijual itu bioengineered ataupun non-fish. So, pada pendapat kamu, adakah kamu setuju atau tidak? Untuk senang-senang saja untuk melepaskan the fish, the bioengineered fish, the market.
Seharusnya nggak senang.
Ya, seharusnya tak senang. Oh, senang. Seharusnya senang atau tak senang?
Tak senang ya. So,
Ya, Tentra. Okay.
I think I have the same opinion on the video that it just have to be some regulation before we release some GMO's product like salmon. In the video, you can see that the salmon already already tested that it is safe to consume. If we have all that GMO products like that, I think it's all right or okay to release. But at the same time, the society will still think like we can still trust it. Like I don't know, is the effect really gonna be safe or not? But if it's our the regulation is already strict, I think it's all right.
Number one, in terms of the we should have the awareness, right? So means that the tight regulation by the government to release that. For example, just only market A, market B, not C, D, and F, right? So selected market that can sell that, so that they are still in control of using the GMO's fish, as compared to the just like release without any, how to say it, any regulation on that, right? And then as mentioned in the video, you can see that they say that their farm, the location of the farm is a far away, right? So it means that in Indiana there is no native salmon. So basically, why? Because if let's say happen, berkemungkinan untuk ikan itu release at the environment susah kerana tiada ikan salmon di situ. So means that if let's say terlepas, itulah mungkin daripada mereka. And then they say that how the nature of the biosecurity that they come up with for to make sure that the fish they harvest and just sell, not release to the environment. Itu yang sangat bahaya. Betul kan? Right? So do you know that how important to conserve the biodiversity, the normal, the original biodiversity? Right? Okay, next. Okay, this one is the how I can say that that we have to concern in terms of genetic modified fish. The number one, GM fish could breed in the wild with native populations of the same species. Can you imagine that the native breed with these GMOs? So they come up with a very, not stable in terms of this ecosystem, right? And then number two, increase the competition of predation. How the outcompete one relative and change the prey relationship. Okay, where might be with the new ecological niche, one species can survive. Because GMOs, we are produced with the additional elements that more superior than the normal one. Right? You watch the Jurassic Park, okay? Where Jurassic Park, they come up with a clone with a new breed of the dinosaur. Kan Jurassic Park, cerita tu. Dah tengok tak? 1, 2, and 3. And how the latest movie is about the dinosaur become as the predator that cannot, which is like a monster, because they have additional characteristic which cannot survive in this ecological, right? And then, of course, GM fish are hard to control and can spread much faster in the wild. So this is why we cannot that happen for genetic modified fish. Okay, next. Right, so this one, do you know about the DNA barcoding? Right, where the DNA barcoding basically for the designated DNA sequence to serve as unique species identifier, and then we use this to compare the sequence variation with differentiate species. For example, in my research study, we have like patin. You can patin? Then like patin buah, patin siam, patin hitam, patin apa lagi di Indonesia? Patin apa lagi? Patin buah, patin hitam, patin siam, patin apa? Jambat. Jambat, yeah. So this patin basically have the same or slightly similar characteristic, right? So we use a DNA barcoding to check, and because they have the unique species identifier, right? Okay, so what happens is that, for example, this, they come up with next application of DNA barcoding where we can use this one next, right? On how this, they come up with the genetic signature of this rare species. They have their own DNA barcoding so that we really know what they claim. For example, that in this market now, they have just only like fillet fish, right? Fillet. So means that we totally don't know what kind of fish is that. We just claim like dory, this is this, this is this. So that we have to come up with a DNA barcode so that we can know that, for example, what did I eat last night? Okay, so everything is in the process packaging, right? Okay, next. Okay, so that they use the fish DNA barcoding kit to start from the start to finish. So means that from here, from the fish, and then the process is all constant, right? Constant until we know that this is from the start, the end, the post-harvest, they all in a good quality of that. So means that if let's say dory, then they go to dory again. Okay, come, have a seat. Alright, okay. Last class, eh? Okay, next. Okay, and then this is what happens that the fish sample we take from the market, for example, for fish fillet, maybe you already know what for example is that final project or something, and from there you extract the genomic DNA, you do the PCR amplification, judge the process, and then you sequence so that you know that whatever the fish that claimed by the manufacturer, that is the fish. To certify, to verify that the same fish that I using, right? Okay, next. Okay, so the second thing by using the environmental DNA for aquatic ecosystem monitoring. So, dia pernah dengar tak? Tidak, in terms of the environmental DNA. Yeah, right? Environmental DNA. Di mana environmental DNA ini contohnya you want to tackle something like reservoir, right? Tasik ataupun sungai yang mana we can use that to detect what kind of this like fish or any organisms that have been there, right? So what you have to do is that apa? Take the water sample, right? Take the water samples because they have given a unit of this microbial gene conservation here, and then you collect the samples from the water, extract DNA, and you can get also from the sediment, and then bring back to the lab, and from there they come up with a species ID of what kind of fish or what kind of this organisms there, because of course they have the DNA there, right? Okay, for example, that okay, we have the indigenous species of patin, which is they call as a patin buah. In Malaysia, it's not easy to get that. And if kita dapat tangkap buah, so 1 kilo might be around 300 ringgit, 1 kilo, which is like 1 million rupiah for 1 kilo of the patin buah. So it's a very, I can say that expensive, which in order the researcher to study, to find out where is the location of the wild species of the patin buah, then we use the eDNA in order to do this monitoring, right? So from there, we do the barcode and then they come up with a barcoding and so on, right? Next. So the same thing, eh? So environmental DNA that offers a new capabilities for marine ecosystem, right? So just take a sample from ponds or ocean, get filtered, and we can get the DNA extracted from the filter membrane. And from there, we do the analysis so we can know that, for example, here, pada musim bunga, the fish is come, but not pada musim tengujung because there is no DNA left there. So this is how we can do for monitoring. Okay, next. Okay, so just now we it's about the environmental monitoring and so on. So we go to the fish health management, biotechnology in fish health management. Again, biotechnological tools such as diagnostic methods, the vaccine, immunostimulants is under development for this health management. Okay, next. Okay, for example, this one. We have the cost-effective disease for shrimp. Okay, on how for the blue stock procedure we can use diagnostic kit to ensure that pathogen-free status for the blue stock. Okay, blue stock is very important because blue stock will come up with this seed and so on, right? And then this technology is a very simple where you can detect 4 shrimp pathogens at the same time. So this is one part of the biotechnology for health management. So basically you can extract from there and then direct they call as a PCR assay to come up with that one. It's easy, it's accurate and allow just only 70 minutes from there. So biasanya kalau di lab di shrimp for the intensive culture they have the lab. So when the broodstock come and then before they send to the hatchery and so on, then they will do this analysis to make sure that it's the pathogen free status, right? Okay, next. Okay, how about this one? Okay, where you also can do the assessment of this bacteria fungus community on the microbiome of the shrimp, right? So from there we start. From there we can do this somehow for the shrimp itself. We go to the image manipulation and then we can do the what we call as a MiSeq sequencing, bioinformatics, so that we can do the data resolution. So the same thing from this shrimp, we can know next. We can know that what kind of these microbiomes, okay, from the different stages in each of that, for example, fungi and also bacteria. So from this, we know that our shrimp, our condition, good or not in the pond, right? Okay, next. Okay, how about this one? DNA vaccine for fish, right? So what it means by DNA vaccine? DNA vaccine. Okay, ini kita ulang kaji balik ya. How to do the cloning. Siapa pernah buat cloning? Siapa pernah belajar recombinant cloning? Ya? Recombinant cloning. Recombinant DNA technology. Cloning technique. Alright. Okay. This one basically adalah kita akan identify. Okay, for the vaccine development, we use antigen from the pathogen. For example, you have the COVID-19 right injection, so they use what we call as the spike protein from the COVID-19, inject to your body, and you have the antibody of the COVID-19, right? So the same thing for the fish. For example, you want to avoid or prevent the disease of the vibrios. So you use the pathogen, for example, the vibrio, okay? And then this is the flagella of the vibrio, right? Flagella, right? So flagella basically can be as an antigen to stimulate the antibodies. So, for example, here we really want to study to become as this one as a dengue vaccine. How to do that? Dengue vaccine for the fish. How to do that? How to do that? Again, get the DNA from the vibrio, right? and then identify the specific gene for this flagella. Betul? So we have this kind we call as a gene encoded for flagella protein, right? Because here is a protein. Okay, that. So how to clone the gene of this protein using the cloning technique? Huh? By PCR to get the gene, right? So this is the PCR example, right? Right. And then you have to clone into this cloning as a cloning vector, betul? Cloning vector here. For example, your antigen here, gene that encode from flagella, for example, is a 700 respect. So 700 respect should be cloned into the cloning vector, right? So the same thing as here, the pathogen, you isolate the gene of the antigenic protein. Antigenic means that able to stimulate the antibody. Okay, that is called antigenic, right? And then after that, you put into the gene of interest of the antigenic protein into the vaccine plasmid, right? And then from there, it will be produced by bacteria, purified the plasmid, and administered to the fish. So basically, what we do is that we transform this cloning with this gene of interest into this E. coli, because it's the simplest cell. So that they have like this, they have like this. What we call the process from this one to put into the E. coli. What is the process? Proses apa itu namanya? Proses apa? Transformation. Nak dengar tak? Transformation. Okay? Transformation where from the cloning vector you want to include in the E. coli that having this, they call as a transformation. So your normal E. coli having the additional gene from the vibrio. Right. Okay, from here, and then again they will produce in a large amount, and then after that they get only this one extraction to get only this cloning vector. So you have like large amount of this one. So many cloning vector in this one, and then after that you inject the fish of this one. So what happens is that when you inject this one to the fish, the gene of interest in the cloning vector, what happens is that the fish will translate into the protein and it will stimulate the antibodies. Right? Do you understand or not? Maybe you don't understand because it's too complicated. But this is my PhD. Right? Okay? So basically, you are dealing with that small antigenic component in the pathogen of the vibrio. From there, identify the gene, transform into the E. coli, come out with there's a large amount of the plasmid that extract, and then you will go back and inject the fish. Right? So this is what we call as a clone a gene to make many copies of it. Okay, so that we can get the same, what's this? The same and large amount of this vibrio gene. This is what happened when I discussed yesterday the insulin. The insulin yang kita inject tu kan, kita tidak extract daripada pig atau daripada lembu kan untuk kita inject, but then we use the technique of recombinant. We identify the gene of insulin, we produce as recombinant, okay, transfer into this E. coli and produces as a recombinant protein. From there, then we inject this to the patient. Right? Ada soalan tidak pasal cloning? Yes? So what what is the difference in silico cloning and cloning? In silico cloning, what do you think about in silico cloning?
First,
So can someone try to Google and read for me what's the in silico cloning? Why this is part of your project? Okay, do you have any idea? Because of course you have idea to ask something that relate to your project or what? Because cloning, yes, we know that. Library construction, yes. In silico cloning and something like okay. No wifi. Pandai kau. Pandai kau. Pun hitung di sana yang itu. Dia tak nak lagi jawapan. Dapat. Kamu ada wifi? Oh, reverse. Yeah, reverse. Right. So it's a reverse transcriptase from the protein to the gene, right? Yes, that one is quite complicated where, negation of the genetic sequence with that, but then basically conclude is only. Right. Right. Okay. Thank you. Any question again? So we move to another one. Yeah, basically it's a human accident. Okay. Boleh ya kamu kalau rasa tidak kamu faham my English, because I'm from Malaysia, I'm not from England. So you can ask me, doktor boleh boleh terangkan kembali dalam bahasa sekian-sekian. Itu kamu faham tak itu? Yang di sebelah presiden, itu kamu yang aku faham? Saya baru kenalkan kepada hari sebelum ini tidak nampak. Kamu ada soalan? Ya kamu ada soalan? Kamu pun serikat juga. Kamu ada serikat? Tidak, kamu serikat. Saya tengok kamu pakai earphone lagi. Kamu ada online meeting ke bersama dengan bisnes apa itu? Saudagar ini ya. Okay, I want to get one question from you. Nanti kamu tanya saya. Kamu tanya saya satu question ya. Satu soalan. Ataupun saya boleh bagi soalan sama kamu, boleh? Okay, alright. Kita proceed. Okay, then next is this one. So, why the vaccine can be used or not? Okay. Number one, they said that risk of autoimmunity because we inject the DNA vaccine direct to the body. So is it any autoimmunity? This is what happened to the COVID-19 vaccine, right? Because now they start to have like side effects on that, right? And then maybe injection site, okay? And then, but basically this new technology can mention that long-term safety issues that remain to be analyzed. Because once you inject the vaccine to the fish, we don't know what happened, what the mechanisms are involved, right? So this is, I think that we need something to look for, regulation again, and also the potential spread of the vaccine to environment, again, something like predatory animals that eat vaccinated fish. For example, that in a pond that having the, some kind of this predatory, and then they just take the fish that vaccinated, and might be that it can effects on this one for the environment, right? Okay, so again, I just put that here, streptococcosis and aeromonasis on how we develop this at the lab. vaccine that having two streptococcuses and pneumonas at the same time, right? Next. And then this is the vaccine. So this vaccine, the inside the pellet is containing the kill pathogen, right? And then we give to the fish. Okay, we give to the fish, we want to see what's the effect of that. And next, so we come out with the paper, this one on expression of immunotranscriptome response. So what I would like to come up with here is that how we analyze the feed-based vaccine by using biotechnology tools. Immunotranscriptome response. Because in the fish, we don't know what happened, right? So kita bagi makan vaccine. So we want to know what happened in the fish body. So that next, we come up with this experiment on the vaccine start first and second booster on the tilapia, and we took the heat blood from there. And next, so we use what we call as a transcriptomic analysis, where we want to study on how gene expression in the body of the tilapia changes when they eat the vaccine. So that is the purpose of the study, right? And why? Because we want to understand the disease, how the vaccine can be respond towards the vaccine, right? Okay, next. So that what they come up with that everything in a pathway that have the impact of the stimulation. So this one ni you tak boleh tahu, tak payah tahu ni tak apa, but I want to say that the red color is basically the pathway, the immune system, right? And it goes to the metabolism and so on, that it's well connected. So by using just only the small samples, okay, of the tissue, we can able to check the gene expression, what happened after they vaccinate with the vaccine, right? On that next. Okay, how about the again, the call as the role of feed additive in enhancing the aquaculture system, because we know that we have a probiotics, we have a prebiotics, we have a symbiotics. What is the difference between probiotics, prebiotics, and symbiotics? Prebiotics, probiotics, and symbiotics. What is the difference between these three? Probiotics, prebiotics, symbiotics. What is this three? What is this? Come up with this three. Come on. You know the back? You know the answer? Yes. No? You don't know? Probiotic. Probiotic. What is a probiotic? Probiotic adalah bakteria bagus, kan? Probiotic adalah bakteria bagus. So they give like live bakteria bagus ke dalam ikan. Jadi ikan akan mungkin mana macam ni kalau minum biogen. Apa nama? Yakult. Yeah, Yakult. You all minum Yakult. So try to give Yakult to the fish. What happens? They have a very good intestine. What is a prebiotic? What is a prebiotic? Prebiotic apa? Mungkin herbal plant ataupun seaweed yang mengandungi compound that can enhance dia punya regulation microbiota in the fish diet, right? And what is a symbiotics? Combination of probiotic and prebiotic. So they have like synergistic effect on these two, right? Boleh semua? So, kan, based on this one, how we use these three in order to come up with a feed ratio, improve growth, enhance immunity, improve water quality, and so on. Right? Okay, next. So, this is what happened when we have the fish with this, the shrimp with this disease. Right? Next. And then in our lab, we try to come up with this called as a Salga lactomix, where we include the lactobacillus and sagasum. So this is also part of the biotechnology to enhance these what we call as genomics, right? Where we found out that it can improve growth performance and also improve survival during uptime because it can enhance the innate immune response and it can protect and modulate the gastrointestinal bacteria community. Maksudnya dari banyak bakteria yang baik di dalam community gastrointestinal. Okay, next. Right, so this is what happened when we mix between the sandasen and also lactobacillus and we mix into the feed, right? Okay, next. So we come up with the, RT-qPCR. qPCR, what's a qPCR? Real-time quantitative PCR, right? Where PCR is just on detection, but real-time basically is to quantify the gene of what you study, right? So we use that and we come up with that A is when we give this with the santalactomix, and B is the control without that. So we know that the enhancement of the feed can improve also the growth rate of the fish and shrimp. So this is part of this biotechnology approach, right? The next. So we published in 2 different papers on the assessment of Lactobacillus plantarum in terms of the growth performance, probiotic pathogen, and also defense, and also how the synergistic of Lactobacillus and sandasen that aided in the diet can improve the survival, disease resistant, immune response, and so on. So all the tools that we use here by using the biotechnology, right? So in our case now, if you can prove in molecular tools, it's easy to publish. It's somehow because that is the current technique. Okay, next. Right. How about this one? Microalgae feed. You know, siapa yang sini study on microalgae? Microalgae, on how this microalgae feed can modify to become as to become as a microalgae that having very good properties by using the nuclear transformation and also proplast transformation, right? So basically, they call as a transgenic microalgae. The normal microalgae, so they also have this genetic modification on the microalgae, right? Okay, we have one YouTube here. Cuba kita tengok apa dia tu. Okay, so tengok betul-betul nanti saya nak tanya soalan dalam right? Okay, boleh? Enaknya makan apa ya? Tempe orek ada yuk. Enaknya pakai bawang.
The food and drink industry produces huge quantities of wastewater, but current methods to treat effluents are often very expensive and not eco-friendly. But under this greenhouse in Italy, researchers are cultivating microalgae that absorb the nutrients for their growth, a technology that could, they believe, meet EU financial and environmental requirements. Campo Rosso, in the northwest of Italy, is part of the European Salt EU project aimed at demonstrating the benefits of algae in wastewater treatment. Across Europe, treated wastewater accounts for around 1% of total emissions. But this scientist is convinced that the process of photosynthesis is the oldest life on Earth and not only produce the extensive treated polluted water to make it a valuable resource.
The microalgae in this project is used to treat water from the dairy industry. This water must first be purified before getting to the second stage, during which it will be used to grow the culture of microalgae you can see in the ponds behind you. After harvesting, we obtain on one side the treated water, which you can see is clear and free of polluting substances. And on the other side, biomass, which is a high added value product that can be sold on the market once it has passed our strict quality control.
The algae first sees the light in this laboratory. This one in particular is spirulina. After 3 billion years of existence, it has many beneficial qualities, including the ability to grow in high salinity water.
This alga is capable of storing contaminants, which are the result of various industrial processes, such as nitrates, which are the main nutrient needed for the algae to grow. The alga is able to store it as protein inside its tissues through photosynthesis, which allows it to increase its concentration in the culture ponds.
Sunlight, carbon dioxide, and wastewater. That's about all these algae need to enable them to consume the pollutants and grow. This self-sufficient process can produce as much as 20 kilograms of dry spirulina per day. The green powder can then be sold to produce cosmetics, fertilizers, animal feed, or even material for 3D printing. An opportunity for manufacturers wanting to benefit from their wastewater. A full-scale test was carried out at this factory in Slovenia, which specializes in animal hides. The water coming out of this warehouse is 3 times more saline than the sea, making it very expensive to treat with conventional methods, which require a lot of oxygen. But this algae culture of salt below produces oxygen for free and in a sustainable way. Here, the algae are mixed with bacteria, absorbing the carbon dioxide it produces and turning it into oxygen.
In principle, we are saving approximately 50 to 70% of energy used in the wastewater treatment. Less energy consumption, less CO2 emissions, and the second very important one, more nutrients are built into the biomass, so we have less problems with eutrophication. We can recycle nutrients instead of releasing them into the environment, producing bad effects into the environment.
Now the research work continues to test various species of algae and make installations like these completely sustainable, so cultures can be built near European factories and pave the way towards a more circular economy.
Alright, so this is microalgae that treat the wastewater. So this is also part of this, what we can say, blue biotechnology, right? Using the microalgae. So basically in Indonesia, what microalgae function untuk apa? Fungsi. Sebagai apa itu? Ya, pakan, kan? But then they can go turn into something that can treat the wastewater, right? So this is microalgae feed, and the next is the okay. This is how this marine aquaculture biotechnology in Taiwan, where they come up with their own R&D in terms of this, all the aquaculture sustainability. Okay. And we can see from biotech, they enhance in terms of this biosecurity management. They have a molecular diagnosis, novel antibiotic agents, vaccine, direct feed microbials, and also biozest systems. And also they have a section of the improved seeds by molecular breeding, using genetic engineering, omics studies, and so on. And for quality, they have like probiotics, bioremediation, and biocontrol. Right? And also for molecular nutrition, nutrigenomics, fish meal alternative, and functional additive. So they have like different kind of this aquaculture sustainability by using the biotechnology, right? Betul tak? In terms of the water quality, in terms of the molecular breeding, in terms of biosecurity management, and in terms of this molecular nutrition. So this is very interesting that covers all for sustainability in Taiwan. Okay, next. Right, and this one in Bangladesh. So they come out with this blue biotechnology too for fish, shrimp, mud crab, mollusk, and seaweed farming, where they divide into 4. So number 1 is how to control and prevent the disease by better pathogen detection, sensitivity, and accuracy by using PCR, molecular dot plot, and so on. And it goes to the supply of broodstock and healthy disease-free post larvae. Okay, they look into this genetic breeding, genetic markers, control breeding and markers, selection of genetic line, and so on. And they go to the lack of quality feed and limited choice, where they develop healthy feed with microencapsulated egg protein. And again, the last one is poor water quality. So it's the same thing in Taiwan. They covered from the water, from the feed, from the disease, and also from the breeding, right? Okay, next. So here, again, to emphasize, according to FAO, the application of biotechnology we can enhance in terms of 4. Number 1, genetic improvement. Number 2, biosecurity and disease control. Number 3, environmental management and bioremediation, especially for water quality. And number 4, for biodiversity conservation, right? And then here I put like three different books here. We have aquaculture biotechnology, aquaculture and fisheries biotechnology, aquaculture genome technologies that you can refer. And that one, the last one case in aquaculture biotechnology basically is like journal, right? For you to refer. Okay? Okay, next. So again, of course, they have an issue for this one. Okay, what is the issue? Aquaculture biotechnology is now evolving, but somehow government needs to support the essential element. Maksudnya apa? They have to come up with a unit or jawatan kuasa, right? To make sure that whatever the products that comes from biotechnology, it must be in control, right? Okay, and then where we can enhance the aquaculture development without any environmental impact. Itu yang nombor satu, environmental impact. Okay, and number two, to eliminate the uncertainties in the risk of biotechnology. At the same time, identify the sustainable biotechnological application in aquaculture and develop according to the risk assessment of the products and methods. What mentioned by your friend just now, we need to have this kind of clear the risk assessment. You know, is it do and don't, something like that, so that we can say that biotechnology can be, you know, a way forward for the future. Yeah, right. Okay, that's all for these topics. All right. So we cover like 4 approach in terms of the biotechnology just now for division, right? So is there any question or we can discuss something for these topics? All right. So siapa ada soalan dulu? Siapa ada soalan dulu? Come on. Okay. Ada soalan? Ada soalan? Before that, kita boleh buat sedikit aktiviti dalam kelas, boleh? Alright. Okay. Okay, you remember just now, right? Kita ada kata tadi ada 4 FAO that come up with something. Apa dia tadi? Kan? 4 bidang of this biotechnology. Biotechnology, aquaculture, right? So they have number 1 on what? Genetic improvement, right? Genetic improvement. Okay, number 2. Okay. Apa ya? Health. Alright, health and biosecurity, right? Health and biosecurity. Security. Okay, number 3. Nutrition. Nutrition, yeah? And nutrition only. Okay, number 4. Conservation, environmental conservation, right? Environmental conservation, betul? Okay, so based on 4 just now, can you give me like contoh and also what is under this improvement, under health, nutrition and environmental conservation? Do you have like macam info kosong kan? Macam tidak kosong ya? Okay, buat paper 5 and senaraikan what do you think from our lecture today? Okay, ada ini ada 3, apa yang kita buat di sini kan? Apa dekat sini tadi? Apa dekat sini tadi? Apa dekat sini tadi? And put your name and your magic number. Dan your magic number. Okay, now 10 minutes. Okay, boleh? Sekarang awak semua orang kena ada info kosong. Now look at like this, and then you can discuss what is information, and from there nanti baru saya nak minta saling depan. Okay, sekarang semua perlu ada. Okay? Do you understand or not? Faham? Puan, faham ya? Nama apa kamu? Faham? Nama apa? Sudah faham ya? Okay. Semua faham di belakang sana namanya apa? Namanya apa kamu? Puan, the first one, nanti akan tulis dekat chat. Okay? Because I don't know why you can see here because it's white. Okay, put that in front. Okay, keluarkan satu ekor paper, yes. Right, apa nama ni tadi? Afina? Elvina. So Elvina akan tulis digit nombor 1, right? Give like a brief whatever that you want, you know about the improvement from my class just now. Right, yang sekarang ini tulis dulu dalam ekor masing-masing and start tulis. Then I will choose for number 2, number 3 and number 4. Okay, come on. Jadi jangan bawa kertasnya. Minta kawan-kawan, siapa ada ekor kosong, boleh pas pada kawan yang tiada. Siapa lagi ada ekor kosong? Ha, ini kawan-kawan banyak. Kamu nak pahala tak? Kamu? Okay, pennya ada. Okay, ada kamu semua ada sini. Alright. Okay, what do we need? Kamu cantik apa nama? Apa nama kamu? Ipan. Ipan, you write on the help and reading. Number 1, Elvina on the general improvement. Number 2 is Ipan. Right. Okay, come on. Boleh yang di belakang sini? Elvina, you can discuss with your friends here. Then write down for the calculation. Do you have an A4 paper? Because you also need to submit to me. Everyone needs to submit to me A4 paper, write down your name and also entry number. Right? And nutrition, you can have someone from here. Yang kamu lah ya. Untuk nutrition, what's your name? Okay, Fauzia, you can come down for the. Here is Fauzia for nutrition. Okay, number 1 is Elvina. Number 2 is Iqbal. Iqbal, you can discuss with the President Aquaculture and Command. You have a very point, yeah? Supaya nanti you can easy to present, right? Okay, the last one, I need satu jejaka. Satu jejaka yang mengenai environmental jejaka. Satu jejaka. Boleh, kalau awak nak nama teks, okay. Nama teks siapa yang awak nak? Kalau teks yang awak. Okay, number 4 is Ramzan and so you have to discuss with your friends and give me the point after this so that I want to know whether you understand or not. Adakah saya punya pengajaran terlampau? Tidak, tapi saya sudah bagi yang mana yang paling mudah dipaham dan sebenarnya. Alright? Okay, silakan. Bukan. You want to try to write down?
Yes.
Now?
Now.
No, give 10 minutes. Discuss with me and then so things get very clear. This one, and this one. Come on.
I don't know.
So you pun, can you do it the same way? Okay.
I will sit over there.
Cari yang sit over. Ramai ni. Hari ni dapat ramai. Okay, 1, 2, 3. Okay, 1, 2, 3. Right. Okay, Okay, can we start with number 2? This number 1. Kemudian Fakultas and itu dah ada jawapan. Okay, Fakultas and itu go first. Come on. Go. Okay, and Fakultas and itu. Okay. Okay, second one. Okay, nampak confident one. Ini original. Tanya saya, kalau kamu belajar di overseas ya, kita dan apa that kind of tapi ini buat tadi. Jadi kalau pada semua kan. Tidak. Kamu biar tak pindah. So you have a section. Yeah, it's Yasin. I think okay, come on. Kita dapatkan from Bali and after that Elvina and the last one is Salwany, right? Okay. Right. Let me give the example. We provide an example of what is GMOs under that, and under that, markers of the because that is the set example of that, right? Okay, because you need us that technique to be concept of manusia. Here, come on, I give you techniques untuk enggak. Contoh ya. Okay, contoh yang di sini. Mau buat ni? Yeah, this one kan eDNA for equity. So from there, you can drop down the eDNA, then you can see that on the water assessment, you can use to get the spacing and you can get the result for it. So we know that. Contohnya di situ katanya betul ke ada yang pakai. So how do we know that it is eDNA, right? This one, kan? Here on how. This is part of this environmental conservation. Right? Okay. You dah dapat jawapannya? PCR, you can use the, but then I want to see some kinds of results. Like this one. It's a multiplex disease detection kit for shrink. That's the answer. Okay. So Iqbal, write down this. Right, okay. Then for the procedure, you put this probiotic in their food fermentation kimchi. Okay, food fermentation kimchi. What is the link with this biotechnology agriculture, Rosia? Okay. Orang okay, sekali Evita, Iwan, and Fauziah with that. And also itu misalnya like high yield manufacturing sites. Right? Yeah. Okay, so in terms of our case, just only that we have a more biotechnology push. Number one, on the environmental condition that I mentioned just now, where in terms of the quality control that include the evaporating, for example, apa? For contohnya processing kan, post harvest. They claim as a fish fillet, which is dory contohnya. How you want to know? You can use a evaporating to check, because at the end of the product is not, it's already in the processed food, right? So that you can check by the evaporating. And also is of environmental data. For example, that we want to check the population of the fish. Is it still there? Is it migrate? And we can use this environmental data, right? Okay, good for this. And then for health and biosecurity, where the vaccine is a part of it. For example, I mentioned about any vaccine just now, and we have a multiplex disease detection kit, and also by health and biosecurity, we can detect antimicrobial resistance, right? In this under health and biosecurity. For nutrition, we have probiotics, symbiotics, growth promoter. Growth promoter means that enhance the production, right? And the last one, for example, we use a genetic improvement. For example, WGS for tilapia genomics, new strain. Kan, tilapia yang kata kononnya hybrid with the fast growth of tilapia from Zimbabwe. And then they start breeding with this propagation. And then we want to understand the genome, then we come up with the WGS, whole genome sequencing, right? And this one is the GMOs where we can push this aqua voltis, and also we use also monitor doping, for example, for biosis, for the improvement, and then here growth promoters and symbiotics under the nutrition, right? Okay, thank you very much. See you again for the next I don't know when. Okay, so thank you very much. So if you have a question you want to ask someone, you can come and
Jadi sesi terakhir dari pertanyaan, kalau teman-teman ada waktu mungkin mau tanya satu silakan. Tapi ya untuk kopinya di sini ini sesi yang terakhir.
Ya. Besok pulang pagi. Ya, saya akan kembali di bulan 7. Kita conference di mana jadi sebenarnya tahun lepas saya sudah ke sini. Saya sudah ke Unai, saya dah ke Bromo, saya pun tak tahu apa itu. Siapa yang belum pernah pergi Bromo? Yang naik tangku naik ke atas tu,
ini sangat ya,
so all the best. And that this is your duty now, right? As a student, so don't give up and always be positive. Okay, always be positive. Right. Okay,
ini mau macam mana ya? Oh iya, iya Pak. Oh,